Okay, so this next part is about cell transport and how stuff moves into and out of the cell. So things that do not require energy, the passive processes are diffusion, osmosis, facilitated diffusion, and filtration. And the ones that do require energy are active transport, endocytosis, exocytosis, and transcytosis. I'll briefly mention transcytosis right now. Transcytosis is when one cell, um, one cell sends material out of it, and the next cell brings it in immediately. And this is how cells can exchange material, or one-way cells can exchange material. So diffusion, movement of molecules or atoms from an area of high concentration to an area of low concentration, doesn't require energy. You may want to get used to using this nomenclature where you say it's a movement of molecules from an area of high, and the brackets mean concentration, to an area of low concentration. Okay, so using brackets for concentration, especially if you're going to take some chem classes, will really help you. So doesn't require any energy. Diffusion, like in this example, is a sugar cube is dropped in water. The molecules break apart and end up being more or less evenly distributed throughout the water. You can also think of diffusion if someone sprays perfume in a room and they as soon as they spray it, immediately the perfume is stronger in one area than the rest of the room, but eventually the perfume will spread out throughout the whole room. Facilitated diffusion just takes material across the cell membrane using a transport protein. So we take one of these transport proteins and we're going to move material through the cell membrane this way. Because these things have a charge, they can't pass through the cell membrane. Okay, so these are water-soluble things. They're still moving from an area of high concentration to an area of low concentration, so it doesn't require any ATP. Osmosis, very specific definition. Osmosis is the movement of water from an area of high concentration to an area of low concentration across a selectively permeable membrane. So the selectively permeable membrane means that only the water can pass through. And whatever the solute is, so if this were, looks like a protein molecule, the proteins can't pass through here because they're too big, but the water can pass through here. And remember, this is water moving from where it's in a high concentration to where it's in a low concentration. So on this side, it looks like initially A has a whole bunch more of these solute molecules. So let's say these solute molecules made up, I'm going to make up a number, 80%. That means water would only be 20% over here. Okay, and over here, it looks like water is, or solute is, the size 30% water, or solute, which would make this side 70% water. So side B has a higher concentration of water, and that's why the water moves to this side through that selectively permeable membrane. Okay, osmotic pressure refers to the ability of osmosis to create enough pressure to move a mass of water. An isotonic solution means it has the same osmotic pressure, so that the cells in an isotonic solution, like this one up here, like A, these cells will just be normal. This is a red blood cell. In a hypertonic solution, meaning that there's more solute concentration outside the cell, which makes there a higher water concentration inside the cell, the water is going to move out of the cell, and you end up with a cell that's all shriveled up like this one. And when you place a cell in a hypotonic solution, meaning that there's less water outside the cell, the water is going to go into the cell, making the cell get very large and possibly burst. Filtration happens, it's diffusion, but it's diffusion created by high pressure. So filtration happens when you have a lot of pressure that's pushing material through a cell membrane or a tissue membrane, like this blood vessel. And this high pressure in here, high blood pressure, is forcing this material out. And that's filtration is diffusion by high pressure. Active transport. Now we get into things that require energy. Active transport requires energy because it's going to move things from an area of low concentration to an area of high concentration. Okay, so this is opposite of diffusion because the material is going from a place where it's forcing against the natural concentration gradient. So in this example, there would be a whole bunch of these little ball things out here, and we're going to force this one little ball all by itself out where there's already a bunch of them. We use active transport a lot to create energy to make our ATP. And active transport, since it's going against the concentration gradient, requires ATP. All right, endocytosis. There's 3 types of endocytosis, but endocytosis itself is how we move large amounts of material into the cell. Penocytosis means cell drinking. And with penocytosis, I'll show you on the next one, but with penocytosis, basically the cell collapses in on itself. So you got the cell like this and it wants to take in some water. So the cell will just collapse in on itself and take in the water. Okay, and it stores it in a vesicle. Phagocytosis means cell eating. And for this one, white blood cells do this a lot. The cell, let's say the cell wants to take in this particle. Maybe it's a bacterium that the cell wants to take in. And this is a white blood cell. The cell actually reaches out and uses these things called pseudopods. Pseudopods, or fake feet, to reach out and grab the material. And eventually the pseudopods close around each other and form the vesicle. Now a lysosome can come along and combine with that pseudopod and digest that material that's in there. Okay, and then endocytosis. This one is showing, looks like the end of that phagocytotic episode. This is receptor-mediated endocytosis, and this one requires these receptors here to combine with some particle, and that tells the cell, hey, we need to bring these particles in. So these purple receptors here are looking for these things. Maybe it's a hormone or maybe it's some nutrient, and they're going to take these into the cell. So receptor-mediated require one of those receptor proteins on the outside of the cell to find whatever it is it's looking for. Exocytosis just is how a cell gets rid of large amounts of stuff. This example says neurotransmitters from nerve cells, but it's also how white blood cells will release pieces of the things it digested during phagocytosis.